Are you, am I in there? You are not connected. Why are you not on here? Here, I'm gonna
end the meeting.
re-invite you. Go ahead. Did it make you a user? Here, I'm gonna resend you this link. Oh, I just sent it to myself. Dang it. Let me. Oh my gosh. Invite. Oh my, I'm starting to panic right now. Ford. Where are we at? How many minutes?
2 minutes.
Go back and try to reshare and see if we can just isolate this. Is there other options in the share?
I share my desktop. Let's see. Because I'm sharing the.
Okay, so try sharing the desktop. But maybe I can. Oh. So Fantini. I'm half Italian, but I know that's not Italian, but. I have my joints.
Okay.
You know, it makes my ears. So it looks like there are a lot of people here because this is a 200-person lecture room, but I only have 20. So they're here because they know this is going to be amazing.
All right, good morning, everyone. So this morning, Dr. Fantini will be talking to us about the nervous system. So she is visiting and is applying for a non-tenure track position, which is primarily a teaching position in our department. So she's going to give a lecture on nervous system. So make sure you're all wide awake and asking and answering all your questions. And I will let her take it from here.
Okay. Do I need to turn the lights off in front or?
Can you guys see the screen well? Good morning. How are we doing? For those of you that like to be antisocial, it's not going to work in the beginning of this class, okay? So what I want you guys to do, it's 9/11. So what I want to do before we start a new chapter, a new material, I'd like for you guys to take a couple minutes, okay? And I want you to think about what you learned about bioenergetics. What you learned last time, right? So you looked at substrates, into energy. So I want you to think about it. And then we're gonna pair you with somebody else and you're gonna talk about what you remember from that lecture. Ready? Set, go.
So we're going to talk about substrate utilization and the cost of
Being antisocial is not gonna work. You gotta get close to somebody.
I know. I love it, but I no too. I tend to favor that side. So I have to. Okay. But I also use lighter background. Yeah, but in the morning, it's not. Yeah.
Hey, this doesn't mean nap time with the lights down.
All right. So why do I like to emphasize my classes? You guys gotta understand that coming here, is it 3 days a week?
Two.
2 days a week for 50 minutes is not gonna do it, right? So the more you revisit what you have learned, the more you think about, the more you talk to people about it, the more likely for you to remember. Okay? So now that you remember most of everything that you learned, we're going to introduce you to a new chapter, and chapter 7 on your textbooks about the nervous system. We're going to talk about the structure and how the nervous system controls movement.
Not working. Okay, cool.
So the objectives of this lecture, again, we're going to look at structure and function of both the central nervous system and the peripheral nervous system. And then we're going to review how we can integrate those systems to make the body works. Okay, so that's known as the sensory motor integration. So if we look at the structure of the nervous system, we divide it into two divisions. Okay, so you have the central and you have the peripheral. So the central, we have our brain and our spinal cord. And then in the peripheral, we're going to have our efferent and afferent division. Okay, I know that is only one letter, but you got to remember this letter, meaning the A and the E, because that's going to make a difference between where the signal is coming from going to. So in other words, if you look at the afferent division, okay, we divide that into 3 parts here. So we have somatic sensory. Every time you hear the word somatic, it has to do with voluntary control. Okay, so is muscle contraction voluntary or involuntary? Somebody. Thank you. So we know that somatic division is information coming from the periphery, the muscles, to the central nervous system, okay? Visceral sensory, think of soft organs, your heart, your lungs, the organs in the gastrointestinal tract, reproductive organs. So again, afferent neurons that are in the visceral part of our body send messages from the visceral parts to brain, spinal cord. Spectral sensory, we're going to talk about them when we're exercising. We're going to talk about muscle spindles. We're going to talk about the importance of motion organ and why they're special. Efferent, now it does the opposite. So we're sending messages from the brain and the spinal cord to the periphery. So again, we know the somatic motor division is going to control voluntary movement. That's the division that controls muscle contraction. And then we have the autonomic motor system, which is involuntary. Okay, and that's controlled by the sympathetic and parasympathetic nervous system. Questions? Good? So then we're going to talk about nerve cells, which are neurons. Okay, this is the most basic structure of the nervous system. And we're going to break down that structure. So we have dendrites. So dendrites are going to receive messages. So it's the intake of messages, and it's going to send the message to the cell body. Okay. And in the cell body, you can see that's where we have the nucleus. And that's where information is processed. Okay. And from there, once information is processed, the neuron is going to decide, okay, do I send this information or not? And then the information is going to be sent out through the axon. Okay. So the axon starts in the axon hillock up here. And then it goes, and we have here end branches, and we have axon terminals. That's the 3 part of the axons there. So that's where it starts at the axon hillock and ends at the terminal. Any questions so far? So one thing about the axon is that it's myelinated. Okay, so I like to use the example of a hairdryer. If you think of the cable of your hairdryer, doesn't have a covering to it. So think of the myelinated sheet as that covering of a wire, okay? And that helps with transmitting the message faster, okay? One of the things that you should note is that it's not continuous. Okay, there's these little spaces in between that's known as the node of Reinvier. And why is that important? Because we can produce what's called saltatory conduction. In other words, we have information jumping from one node of Reinvier to the next, which makes conduction faster. Are you with me? Okay. One other thing that you should know is that the axon can be as small as 1 millimeter to as long as 1 meter. Okay, so I can have a alpha motor neuron, which is a neuron that innervate muscle fibers. So they're the neurons that make our muscles to contract, and I can have the body of the neuron located in my spine, my lumbar spine, and the axon goes all the way to my toes. Okay. Any questions about our wonderful basic structure of the nervous system? Oh, I like to look at my notes so I don't forget anything. Anyone have heard of multiple sclerosis before? So it's an immune disease that attacks the myelin sheet. It damages the myelin sheet. And if we think of information being transported from point A to point B, that information gets lost, and folks with multiple sclerosis are going to experience fatigue, loss of motor control, depression, okay? And it's just the fact that we're not as efficient anymore transmitting messages from the brain and the brain and the spinal cord into the rest of the body, okay? Questions? So the way that the nervous system communicates via neurons is electrical communication, okay? So neurons are known as excitable tissues, okay? Because they have the properties of irritability and conductivity. So irritability will be they respond to stimulus, and conductivity is the ability of conducting the electrical stimulus. One of the things that you noticed on this figure here that's from your text is that you see the inside of cells are more negative than the outside of cells, okay? And when we talk about what determines that, we look at ions, okay? And the permeability of the plasma to those different ions, and the concentration of the ions is going to determine the resting membrane potential of a cell. For instance, a muscle cell is approximately right around -70. Okay, but there is a average, okay, of negativity within different cells. And what maintains resting membrane potential of a cell is going to be the sodium potassium pump that you see here. So these are channels that allows, so besides the permeability of, again, the cell membrane here, we have these channels that allow sodium and potassium to go in and out of the cell, okay? But to maintain the resting membrane potential, we use the sodium potassium channel. Have you guys heard of the sodium potassium channel before? So if you look here, there are three, there's space for 3 molecules of sodium go into the cell and space for 2 molecules of potassium to go out of the cell. Questions? I have 2 pictures here to help you understand. So let's talk about the muscle cell. Let me go on this side now, because I'm showing favoritism to you guys. So muscle cell minus negative, like I told you. So there is a stimulus. So we want to contract the muscle. It's a voluntary thing. I want to start walking. I want to stand up from the chair. I want to move my fingers to right. There's a stimulus. Okay. So with that stimulus, if the stimulus is strong enough, which we're gonna talk about it here in a little bit, what does that mean? The channels for sodium are going to open. Is sodium a positive or negative ion? It's in there. Positive. So the whole idea here is to make the inside of the cell less negative. The way to do that is to push positive ions into the cell. So the channels for sodium open. Sodium rushes into the cell, depolarizing the cell. So depolarization is a word you should know. Might be on your exam. So when we depolarize the cell, we're making the cell less negative. Okay, and you see here the actual potential being created. So on our way up, this is when sodium channels are open, sodium is rushing into the cell. And then when we get to about 30 positive, those channels close and we have to repolarize the cell, which means we need to go back to make the cell less negative and back to where it was at the resting level. Okay, so during repolarization, where you see this going up or going down and making the cell less negative, the channels of sodium are closed. Now we open potassium channels and potassium runs out of the cell. Potassium is also a positive ion. And then as we're going down here, you see that we go below where it was at rest. This is called hyperpolarization. And there is a little leak of potassium that continues to go out. During the beginning of the hyperpolarization here, as we're going back into that resting potential, the cell is not very likely to be activated again. Okay, so that specific alpha motor neuron that's contracting a certain number of muscle fibers, it's harder to activate that same alpha motor neuron in the very beginning of that ascent to go back to the resting potential. Questions? So I told you guys, you know, how much stimulus do we need? Okay. So here on this photo here, you see a alpha motor neuron, and you see the alpha motor neuron innervates different muscle fibers. Okay. So in order for the muscle fiber to contract, we need an actual potential to arrive at the neuromuscular junction. A graded potential is just a little stimulus that does not create an actual potential. In order for an actual potential to happen, we have to hit approximately negative 55 millivolts. Remember the cell, what was the muscle cell at rest? Minus 70. So we need to stimulate that cell enough to get to approximately 55. If we don't get there, all we have is a graded potential. So it's a little stimulus. When we get to approximately minus 55, we're able to see the actual potential. When that actual potential travels down the axon, so remember, actual potential is only going in one direction, dendrite, cell body, axon. When an actual potential is created and it travels down that axon, that all or known law or all or known response means every muscle fiber that's innervated by that alpha motor neuron is going to contract. So either all of the fibers contract, or if there's not an actual potential that arrives at the neuromuscular junction, none of the fibers that are innervated by that specific alpha motor neuron will contract. Questions? So if we look at synaptic transmission, okay, it's just the transmission of information between a presynaptic neuron, so the neuron sending the message, and the postsynaptic neuron, which is the neuron receiving the message. Okay, and the interesting thing with that transmission is that we go from an electrical signal to a chemical signal and back to an electrical signal. So you guys remember I was talking about the parts of the axon, and there are 3. Where did it start? What's that? It started with an H. Come on, go back to your notes. Where does an axon start? Did you look right? And then I talked about 2 other things that the neuron had, or the axon had. Branches and the terminal. So the terminal is at the very end, and in the terminal we have neurotransmitters. And once the actual potential arrives at the terminal, those neurotransmitters, which is a chemical, such as acetylcholine that you're gonna learn, that's involved in muscle contraction, that's released. Okay, and the attachment of that neurotransmitter that's sent from the presynaptic neuron to the postsynaptic neuron is what's going to start a new action potential. So again, we go from electrical to chemical back to electrical. Questions? So of course, in order for an electrical signal communication takes place, there has to be a stimulus. That stimulus has to be propagated, okay, out of the axon of one neuron and into the dendrites of the next. Has to be transmitted from one to the next. Questions? You guys are very quiet. You're not going to be quiet here in a minute because I'm going to make you work. So here's an example of, again, this is the terminal. Remember I was talking about the terminal of the axon? You see these little vesicles? That's where the neurotransmitters are located, okay? And like I explained it to you, the neurotransmitters are going to bind to receptors on the postsynaptic cleft here of the postsynaptic neuron or muscle cell membrane. Okay, we're talking communication between a neuron and muscle fibers. And remember, where in the neuron is information processed? Don't be shy. If you get it wrong, we can get it right and you're going to remember. The nucleus. Okay? So the nucleus is going to, so information is coming down the dendrites into the nucleus. The nucleus is going to process that information. And once that information is processed, the nucleus decides if it's going to continue to send the information down to the next neuron, or if it's going to inhibit, stop that message. Okay, so if we allow that electrical signal to continue, we have an excitatory postsynaptic potential. Again, a message that's going to continue, that's going to be delivered, that's going to make your muscle contract is known as EPSP. And we have temporal and spatial. So temporal is the message coming from one single neuron. Spatial will be message coming from different neurons to like the same muscle that of course are going to attach different fibers. Okay. Now, if the nucleus decide the message should not go on, we're going to experience what's known as inhibitory postsynaptic potential. So instead of releasing neurotransmitters that are going to excite the muscle fiber to contract, we send neurotransmitters that's actually going to inhibit the muscle from contracting. So therefore, there's no muscle contraction. So again, signals can be excitatory or inhibitory. Are you ready to work? I told you you're going to work. Before you go into this, I want to go back. What's part of, quick review here, what's part of the central nervous system? Brain, spinal cord. What's part of the peripheral division? The afferent and efferent sensory neurons. Okay, which one of those send messages from the body to the brain spinal cord? Afferent, which then is the efferent, that's the one sending messages from the brain and spinal cord into the periphery. Good job. What are the three parts? Well, let me start with this. What is the most basic structure of the nervous system? The neuron, right? Also known as nerve cell. Okay. What are the three parts that we talked about? What receives messages? Dendrites send message to the before the axon. The cell body, and then from the cell body up to the axon. Do actual potentials go different ways, or it only travels from the dendrites to the cell body out to the axon? One way. Good job. Now, what I want you to do, I want you to get together with a classmate or on your own. I want you to put this in order. And this is the process of depolarizing a cell and creating an actual potential. If you did it by yourself, pick somebody else's brain and look at if it matches or doesn't. Are we ready? No? Okay. You guys can celebrate and go get coffee if you get this right, okay? After class. Are you ready? All right, what's the first thing that happens? Thank you. Main brain potential is at resting state. There's no stimulus. The muscle is not contracting. And I'm referring back to the muscle because of this is exercise physiology, right? But we can talk about communication of, again, neuron to neuron, neuron to different organs within the body, different cells. All right, so then what happens? The depolarizing stimulus is received. So remember, we're depolarizing the cell. What is next? So follow the word. We're depolarizing the cell. Membrane depolarizes to threshold. So remember, and you remember what the threshold is, you have to be approximately at what to create an actual potential? -7 is where we start, 55. Okay. So then what we see is sodium channels open and sodium enters the cell. When sodium enters the cell, what does it do to the cell? It makes it less or more negative? Less negative. So then we have sodium channels closed because we're ready to do depolarization, right? We depolarize the cell. So then the sodium channels are going to close. And in the process of repolarization, we open there it is. Sodium channels close and it's lower sodium channels open, or excuse me, potassium channels open. So now we're yes. So step 4 is, sorry. So we went from membrane potential is in resting state to depolarizing stimulus received to membrane depolarized to threshold. Everybody good? And then we have rapid, sodium entering the cell, which is right here. That's step 4. Step 5, now we have those sodium channels closing. And the opening of potassium channels. Potassium moves in or out of the cell? So potassium is, let's start with this, is a positive or negative ion? Positive. If the job, or if we're trying to achieve, is to make the cell again less negative, are we moving that out or in? Out. So then we have potassium moves from cell to extracellular fluid. That's step 6. And then step 7, the potassium channels remain open. And additional sodium or potassium leaves the cell, hyperpolarizing it. Remember, we go below the -7, the -70. There's a hyperpolarization of the cell. And then the last step is cell returns to resting, ion permeability, and resting membrane potential. How was it? Again, the more you guys practice things, because if I was just standing here and talking to you about this will help you to retain that. So go through the process. Every time you learn a process in the course, go back and try to do it yourself. Any questions on this before we move on? So we talked about the basic structure of the nervous system. Now we're going to break down central versus peripheral, okay? So central nervous system, we have our beautiful brain here. We're going to start there. And I want you to notice the 4 major divisions of the brain. So you look at the encephalon, the cerebrum, cerebellum, and the brainstem. So those are the 4 major divisions of the brain. Okay. Now, instead of standing here and talking to you about which one of those divisions or part of the brain does what, I want you to dig into your own brains and use either previous knowledge or talk to someone beside you and try to figure out which part of the brain controls what part of movement, what part of the body. Ready, set, go. I like to make you guys work. So what does what? All right, you're getting quiet. You guys ready? Yes? Okay. So which one of those serves as a major sensory relay center? Again, if you get it wrong, we're just going to learn together. Anybody? What's that? Nope. Nope. The thalamus. Yes. So the thalamus serves as the major sensory relay center in the brain. What consciously controls skeletal muscle movement? Nope. What's that? You're going there? The motor what? Yes. Good. The motor.
But you said cerebrum, is motor cortex a part of the cerebrum?
What's that?
Like, is motor cortex a part of the cerebrum?
Yeah, it's part of the primary motor cortex. So it's the frontal lobe. Okay. So if you guys see, so you're gonna have motor cortex, thalamus, hypothalamus as your answers. Okay, so the motor cortex is responsible for conscious control of skeletal muscle movement. Okay, and I want to make sure you guys get all the answers, and we're running out of time here in a little bit. So maintaining homeostasis regulates internal environment. Hypothalamus, it's huge when we talk about helping us to continue to exercise. And then we go to coordinate skeletal muscle function and tone and control cardiovascular and respiratory function. Brainstem. And then you had control rapid, complex movements. We have 2 left. Pick one. Cerebellum. And then the last one here, which is initiation of sustained or repetitive movements, such as walking, running, posture, muscle tone, basal ganglia. Okay. Yes, ma'am.
I've already asked this, but I'm always confused. This is cerebrum. Is this like a, like an umbrella term under all this?
So, no. So the cerebrum is one of the, so that's where you have your lobes. So you have, right? So the frontal lobe is where motor cortex, primary motor cortex is housed in the frontal lobe. And that's what, that's the part of the brain that controls consciously skeletal movement. Does that make sense? Any last minute questions? Because you have 20 seconds. All right, so the next part then, what is the other part of the central nervous system? The spinal cord. So we'll continue. I don't know, your professor will continue. We'll talk about the, again, spinal cord next, and then you go into the peripheral system. Okay? No questions, comments? Remember, for those of you that got the answers right on the previous one, go get each other some coffee. Okay? Have a good one. Stay out of trouble.